Welcome to the 2020 Seattle Chapter ARCS Foundation annual event. My name is Susan Potts, and I am honored to say that I have been a member of ARCS for the past 22 years. The goal of this event is to showcase the remarkable young scientists at the University of Washington and Washington State University, and to offer you a glimpse into the profound and vital work being accomplished by this talented group. We thank you for attending and for your ongoing commitment to ARCS. ARCS Foundation is a national organization of women whose sole mission is to advance science in America. Seattle Chapter ARCS Foundation fellowships are a strategic tool used to recruit world-class graduate students in 48 disciplines across the areas of science, engineering, and medicine. These fellowships allow our two premier universities to compete with peer institutions across the country and to attract highly qualified students and bring them to the state of Washington. Statistically, many of these students will complete their education and then choose to remain in Washington State to pursue their careers in industry or academia. We are proud that the Seattle Chapter ARCS Foundation has given over $20 million cumulatively to this effort over the past 42 years. Nationally, the ARCS organization has given over $120 million to approximately 11,000 students over its 62-year history. The ARCS Foundation is one of the largest contributors to bench science education of any private membership organization in the United States. ARCS Fellows are looking to science to solve the most critical problems facing our planet today. Challenges such as climate change, agricultural sustainability, and global health issues. ARCS members, supporters, and our industry and foundation partners are committed to providing targeted support for basic research. We believe it is a national priority. This support is provided through our consistent funding of outstanding students at the UW and WSU. These world-class scholars come to the state of Washington to pursue their individual scientific research, but there is a secondary benefit to having them, a trickle-down effect. Nearly all of ARCS Fellows teach undergraduate classes while they are pursuing their degrees. This gives undergraduates the opportunity to be taught and mentored by these involved and passionate graduate students, perhaps even inspiring a whole new generation of STEM scientists. We will hear firsthand from several of these outstanding fellows in a few minutes, but first, I would like to introduce Sarah Watts, the president of the Seattle Chapter of ARCS Foundation.
Hello, I'm Sarah Watts, president of the Seattle Chapter. We're delighted to have you with us today to honor our commitment to scientific achievement and to recognize the impact of your commitment to ARCS. This year, Seattle Chapter ARCS Foundation funded 180 fellowships at the University of Washington and Washington State University. These efforts go beyond the university labs and classrooms, and we could not have awarded these fellowships without our inspiring members and donors who give generously each year. We're especially grateful to our fellowship and endowed fellowship donors and our corporate and foundation partners. Many of their logos and names you read at the beginning of our presentation are familiar to us all because of their recognized commitment to scientific philanthropy. We are proud to be investing in scientific progress with each of you, and thank you for partnering with us in our ongoing mission. I would like to salute the ARCS scholars and alumni nationwide who are actively engaged in COVID research. We are deeply appreciative of your creative and groundbreaking collaboration across numerous disciplines and entities to address this terrible pandemic. And now back to you, Susan.
Each of the four scholars you will be hearing from today speak to the importance of funding scientific advancement. You will also learn about the tremendous impact your financial generosity and institutional support is having across our statewide ecosystem of academia, scientists, and industry. Our first speaker is Sanjay Srivatsan. Sanjay is a fourth-year MD PhD student in the Department of Genome Sciences at the University of Washington. Sanjay has recently pivoted his research. He was focused on genome sequencing and creating a 3D map of how cells form in the body, but is now working to develop a testing strategy with the Seattle Coronavirus Action Network. The goal is to provide COVID testing throughout the Seattle area. Sanjay will be speaking with us from the University of Washington campus.
So we're in a world surrounded by technology, and that goes from cars to machines to computers. But the kind of technology that I'm really interested in and what I work on is biotechnology. And the idea behind biotechnology is how do we take things that are naturally found in life and use them to solve problems in our world. So I first developed this love for biotechnology at the University of California, Berkeley, where I was an undergrad. There I learned from world specialists like Jennifer Doudna, who invented the CRISPR-Cas9 genome editing technology, how to think about biology in this different way, how to think about it as a parts list that can go together and make something new. After my time at UC Berkeley, I went to the Vaccine Research Center at the National Institute of Health. There I did a 2-year fellowship thinking about how we can use structural biology, the shapes of proteins, to actually develop vaccines against HIV and RSV. So that same approach by that same group is actually what's going on right now in the development of a COVID-19 vaccine. And that's currently under phase 3 clinical trials with Moderna. And I'm looking forward to them making great progress on that front. After finishing my time at the Vaccine Research Center, I looked to do an MD PhD program. And I was looking between a number of different schools, Yale in New Haven, Rockefeller in New York City, Emory in Atlanta, UCLA in Los Angeles, and the University of Washington in Seattle. What drew me to Seattle specifically was the ARCS program. And in the ARCS program, I saw a community of people that actually cared about science, technology, and the actual scientists that did that work. That willingness to actually fund people directly actually goes a long way in building confidence and telling someone that this community actually cares about them. So my donor, Molly Pengra, and I actually connected on more than just that. So she and I actually have very similar interests in terms of science and research. She's looking to make cardiomyocytes from stem cells. And as you'll see, my work actually ties into that goal directly. So when I came to the University of Washington, I looked around and I had the chance to pick any department I wanted. The one I chose was genome sciences. The reason I chose genome sciences is that it's a small institution, small department, where large things have happened and are happening right now. For example, one of these things is that human genome was pioneered and a lot of it was sequenced at the University of Washington in the Department of Genome Sciences. So I draw inspiration from that large kind of paradigm shifting work in my own work, in my own day-to-day, when I imagine what I can do to create new technologies or measure new things. What I do is I work in the labs of Cole Trapnell and Jay Shinduri on something called single-cell sequencing. So I've told you about the human genome, which is all of the DNA in every cell, but every cell actually expresses subsets of the genome as genes. And what I do is I take a single cell and measure which genes that cell is expressing. And this is important because we're all made up of a bunch of different cell types, which are all expressing different genes. And by discovering what genes each of the cells are expressing, we can understand how they came to be and how diseases start to occur. One specific project that I'm very excited about right now is building a 3D map of how organisms develop at a single cell resolution. What this means is that as an organism develops, my vision one day is to have data that wherein you can point to a single cell and ask what genes that cell is expressing and what it's going to become over time. And I think this is important for a couple different reasons. One, it'll tell us how we come to be from a single cell, kind of our origin story. And the second thing is that it'll give us that map to actually take a stem cell and turn it into any cell type you want, like a cardiomyocyte. So this is what I usually think about, this is what I usually do, but 6 to 9 months ago, this all came to a grinding halt with the SARS-CoV-2 pandemic. And at that time, what I realized is that many of the same skills that I use to do single-cell sequencing could actually be used towards coronavirus testing. So I joined something called SCAN, the Seattle Coronavirus Action Network, which is a partnership between the University of Washington and Seattle Children's and King County Public Health. And what SCAN does is that they ship kits out to individuals in the community and test those kits for coronavirus, influenza, and RSV, and send those results back the next day. And what that does is it gives you a sense of how the pandemic is changing at the community level. And we're learning things about how pandemics start and spread and how to measure them, which we can use for years to come. One really interesting thing about SCAN is that we detected the first community case of coronavirus in the United States. So this effort is really important and dear to my heart. Finally, I really couldn't be here without the network of mentors and peers that are here in Seattle. So Seattle's a community of people that care about technology, they care about science, and that's embodied by my mentors, Cole Trapnell and Jay Shinduri. They are people that I looked up to as scientists, as parents, as citizens, as individuals, and I feel fortunate that I can learn from them and draw energy to kind of push my own work forward. I think that supporting scientists and individuals like that is one of the greatest gifts, and I'm thankful to ARCS for doing exactly that.
Our second speaker, Doan Hoxson, is a second-year ARCS Fellow at Washington State University in the School of Entomology. Doan will be speaking to us from a Washington State University pear orchard, similar to sites where she gathers insects for lab observations. Doan is committed to minimizing the negative environmental impact of pesticide usage in agriculture. Please listen carefully as she's going to allow us to hear actual insects sing.
Hi, my name is Doan Hoxson, and I'm a third-year fellow from WSU in the Department of Entomology. You can say that I've been studying bugs since I was 7, when my boy cousins and I got into spider fighting. We kept these spiders in matchboxes and used barbecue skewers as platforms for the fights, where obviously my spiders were always winners. I grew up in a little island called Saipan, which is a U.S. territory in the Pacific Ocean. When I was growing up, I really wanted to be a lawyer or a doctor, but found that I loved learning about nature more. So when my high school biology teacher encouraged me to take AP Bio, that's when I knew I wanted to be a scientist. And I picked an ecological field because I get to be outside, observing, learning, and listening. I started as a pre-med in St. Louis University, but found myself working in a pollinator biology lab, where my interest shifted from just plants to plant-insect interaction. And from there, my ARCS fellowship allowed me to drive all the way across the country to WSU to live my lifelong dream of studying bugs. I am the 7th ARCS fellow in my mentor's lab, and he was previously also an ARCS fellow himself, so he understands the support and community the ARCS fellowship provides a scholar. As an ARCS Foundation Seattle chapter fellow, I am gratefully experiencing the warmth of this community. So thank you, ARCS Seattle chapter, for your encouragement, support, and kindness. My field of entomology is gaining interest as we become more aware of the negative effects of pesticides. My research is part of a growing field in the integrated pest management that develops strategies to lessen and hopefully eliminate the use for chemical pesticides. My career is devoted to finding mating disruption tools that uses airborne acoustics or vibrations produced on plant stems and leaves so that we may break up the mating behavior and so that, less bugs are born. My specific research focuses on the reproductive processes of an insect that is a pest of pear called the pear scilla. These insects damage the fruits by pooping out a sugary substance called honeydew, which actually recruits mold and makes the pears unmarketable. But because of COVID-19, my research has become more bench science because I can't go out and collect insects from the orchards or interact with farmers on field days. But I do have collaborators from Wenatchee and Yakima that send me bugs through the mail. And when I receive these bugs, I aspirate them into a vial and put them in plants in the greenhouse, where I can observe and record what they're doing. What I'm interested in is how they sound like and what makes them sing, because that's part of the mating behavior. So this is what a male pear scilla sounds like. And if we know how they behave to reproduce, then we can disrupt that behavior so that they can reproduce less. My greenhouse experiment is essentially a small-scale orchard where I can audio playback vibrations through the stem to overwhelm the senses of the insects. And then I will count how many offspring they produce and compare that to insects that did not get vibrational playbacks. And if we see a difference, then we can scale up to an orchard-wide experiment. And I'd like to thank the Pear Commission and the Hyde Planned Giving Endowment for supporting my research so far. Among other important benefits, decreasing the use of pesticides will significantly cut overhead costs for farmers. An article I read about mating disruption strategies projected that these strategies will cost $56 less per acre of pears compared to using pesticides. So if we have 35,600 acres of pears in the Pacific Northwest and the Palouse, that's $2 million that farmers could save annually on pest control. And I believe that this type of cost savings will follow a similar trend for other types of mating disruption. We're not trying to wipe out a whole insect population, but we are trying to reduce that population so it's easier to manage with the tools that we have. And if I'm not playing with bugs in the lab, I love going out and teaching kids about bugs. As part of the Entomology Graduate Student Association, I take live bugs from our museum and take them to insect expos and outreach shows. And I love seeing kids go from not wanting to touch the bugs to not wanting to leave them. But because of what's going on right now, we had to cancel our expos and shows, so I'm sewing face masks in my spare time. As we go towards more sustainable agriculture and a way of life, we are becoming more aware of the negative impacts of pesticides use. My research will help farmers control pests, but also decrease the negative environmental impact that pesticides have. We must mitigate the mass extinction of many animals that are affected by pesticides, whose disappearance would have a negative impact on humans. My vision for a healthier planet is that by reducing pesticides, we can reduce the production of unnecessary carbon dioxide and other greenhouse gases. It is important for us to make this commitment to scientific research now, as our timeline for global health has been greatly shortened. So thank you for believing in ARCS and the power of science.
Nick Maurice, our third scholar, is in his fifth year PhD work at the University of Washington in molecular and cellular biology. His current research is focused on how inflammation shapes T-cell functions, and is currently working as a volunteer to establish protocols for clinical trials on COVID. He will be speaking to us from the University of Washington campus.
Hi, I'm Nicholas Maurice. I am a Seattle area native, actually growing up in the suburb of Woodinville, Washington, where I attended high school at Woodinville High, before making the move to the city to attend Seattle Pacific University for my undergraduate degrees in physiology and chemistry. I'm now here in Seattle pursuing my PhD through the molecular and cellular biology, or MCB, program that's jointly offered between the University of Washington and the Fred Hutchinson Cancer Research Center. And I really found my passion for research starting in my baccalaureate, where I was trying to manipulate the machinery of a cell, but this sort of grew into a passion for immunology and trying to understand how we can manipulate immunity as therapeutic. So I first started as a post-bac technician at the HIV Vaccine Trials Network, where I was really focusing on how T cells, which are our pathogen-specific immune cells that have memory, are generated in response to various HIV vaccine candidates. Since pursuing my PhD, this has sort of changed the lens of my research to focus on more of the cues that can actually dictate the fates of these T cells via things called inflammation. And while we might think of inflammation as swelling or tenderness in the context of infection, inflammation is actually from a wide range of molecular signals that cells use to communicate with one another. And in my PhD, what we've been really focusing on is how inflammation impacts cells that are not specific for an infection that we're seeing. So when we think of a T cell, it's really constrained by which infection it's been raised against. So we think of a flu-specific T cell as really only responding to flu. and that it would be what we call a bystander or inert during a different infection, let's say a bacterial infection with Listeria. And what our group has shown is that while convention says that these cells are inert, that's not the case. These general inflammatory cues will actually activate bystander T cells to mount a rapid immune response, to be the first responders, actually, rather than a inert one-trick pony. And although we think of immunity as something that we want to be really proactive during infection, it's really something that has to be balanced on a scale where we want to clear pathogen, but we also want to mitigate any sort of autoimmunity or self-tissue damage that might occur. So my research right now is focusing on how we can sort of restore the balances to these scales. In one hand, we want to attenuate or limit these bystanders in vaccination because we think that they might actually impair the vaccine-specific response that occurs. And this is particularly important in elderly populations because as one ages, the pool of T cells that expands can become more and more bystander activated. And if we could potentially inhibit this bystander T cell phenomenon, we might be able to improve the aged response to a flu vaccine. On the other hand, I actually got a 6-year National Cancer Institute grant to investigate how we can accelerate these bystander T cells in order to leverage them as an anti-tumor immunotherapy, which might actually synergize with other pre-existing standard of care tumor therapies that we have on the market. I had a awesome array of offers from various institutes all over the world, and the wealth of resources at the U paired with the support from ARCS really drew me to come here. So while ARCS funding might feel like it's only supporting a graduate student, its scope is actually much more far-reaching. In my career, I've actually mentored both undergraduates and high school students from the Seattle area, including local high schools, the University of Washington, and Seattle Pacific University. And here I've really worked to mentor students not only to become better scientists, but to figure out which kind of career trajectory that they want so they can implement change on their own terms. I want to highlight that in this sort of post-COVID world, that pursuing graduate school is a little bit more tenuous in the sense that all of our interviews for our program are going to be done digitally, which doesn't offer the benefit of showing off the wealth of resources that we have here at the University of Washington through the Fred Hutch and also through ARCS support. And I think that it's going to be absolutely critical to have this funding and support from ARCS to incentivize bringing the best students that might be dissuaded from starting their PhD program by this current pandemic. And this is absolutely critical because graduate students are the ones that are going to not only be developing new ideas, but promoting these ideas that lead to new business strategies and new things that might actually go on the marketplace as a therapy. And that's important for building business relationships within the Seattle area, as well as extending to the fact that we're also educators to some degree and actually leading to a greater understanding of science that's occurring in the greater Seattle area. While COVID has completely reshaped our day-to-day lives, this pandemic has also reshaped how we conduct science, where we have to socially distance within the lab. And this really changes, I think, how grad students are going to enter their programs because they're going to be more reluctant since they can't necessarily conduct the science that they're so interested in. And this ARCS funding is going to be absolutely critical to incentivize bringing these students in to begin their PhD studies. And these studies, while we might think of them as essential for advancing our scientific knowledge, they're also really important to move new therapies into the marketplace, to build business partnerships within Seattle, to also help foster education, since us grad students are part-time educators at the universities. We're mentoring these upcoming students that are going to take the baton from us. So this funding is absolutely critical to give this sort of security in an otherwise unpredictable time.
Our fourth ARCS Fellowship recipient, Brian Lee, is a distinguished investigator at the Allen Institute for Brain Science in Seattle. He is currently classifying cell types in the mouse and human brain. Brian will be speaking with us about his work in creating a global resource to guide and facilitate research on numerous topics, including Parkinson's, Alzheimer's, and depression. Brian will be speaking with us from South Lake Union.
My name is Brian Lee, and I was an ARCS Fellow in 2007. I was interested in psychology as an undergrad, and I was deciding on whether to pursue a degree in counseling. And in doing that, I decided to become a volunteer crisis counselor for the Suicide Prevention and Crisis Line. While I found that role fulfilling and I enjoyed helping others, I became more interested in what was going on in the brain as opposed to treating them. So I decided to pursue an advanced degree in neuroscience. And with going to graduate school, there's always this uncertainty about the financial stability of it. In my pursuit of which school to decide to go to, I was awarded an ARCS Fellowship with WSU. So the financial uncertainty of graduate school is daunting for anyone, and the ARCS Fellowship that was provided with the acceptance of going to WSU helped me solidify my choice for attending that university. Once attending WSU, I joined the lab of Yan Dong, who is now at the University of Pittsburgh, and he is the one who really taught me how to become an excellent scientist. He taught me to be ethical, and he taught me to always strive for more. Due to his strength and motivation and the ARCS Fellowship provided by Sally Benke, I was able to obtain highly sought-after positions for my postdoc and scientist career. So for my postdoc, I worked at the Scripps Research Institute in Florida, where I continued to look at the neural circuitry of addiction-related behaviors. After completing my postdoc, I moved up to Seattle to become a scientist at the Allen Institute for Brain Science. So if you think of the brain as a computer, we know all the components of the computer and what makes it run. In order to understand thought and cognition, we need to understand all of its parts. And that is exactly what we're doing at the Allen Institute for Brain Science. We are trying to understand how the individual components of the brain contribute to computation and cognition. So right now, we are working on classifying cell types in the mouse and human brain. And to do that, we've been perfecting an advanced technique that allows us to gather multiple modalities of data, the electrophysiological type, the shape of the neuron, and its genetic profile from one single cell. This data allows us to capture the most accurate profile of a single neuron. Then we can take these profiles and put them on our online database to allow other researchers around the world to use to help facilitate their research, whether it's depression, Alzheimer's, or Parkinson's. Our vision is to accelerate the pace of scientific discovery and nurture medical breakthroughs. Our online database, which we can consider an encyclopedia of cell types, attracts over 500,000 people a year with continued growth. So without these resources, many labs would be set back many years collecting the same data that we could collect in the fraction amount of time. All of this is possible because Paul Allen has the vision and foresight to advance science 12 years ago with donating over more than $750 million. When I speak at international and domestic conferences, more than 90% of the people are aware of the Allen Institute and what our mission is. I believe that understanding the language of data and science is more important now than it ever has been before. I think it's important that we teach all generations how to understand the data and make data-driven decisions. In the past and present, we have used data and science to make important discoveries and advancements that have changed and can change the world. So some of the challenges that we face is learning to accept the data for face value, who's going to interpret the data, and how do we apply this to certain situations in society. If we are making data-driven decisions and respecting the scientific process, then we should be making the appropriate and right decisions. So here at the Allen Institute, we have the unique advantage to foster these collaborations, whether it's the work with the Allen Institute for Immunology and providing the framework for Nick and his work with T-cells, or whether it's the Allen Institute for Cell Science and helping Sanjay with his work on 3D maps. So without all of you as ARCS donors, your encouragement and desire to move science forward, we would not be here today, and I would not be here speaking to you today, and I thank you for that. Please continue to promote science through your generosity as members and friends of the ARCS community.
Thank you all for joining us again. We just have a couple more comments that we would like to have you make before we end this discussion. I'd first like to thank you again for inspiring us with your research and commitment to our scientific readiness. We are talking today about advancing science and why scientific progress matters more now than ever. To conclude your remarks, would each of you answer 2 questions for us? First, why does scientific progress matter? And secondly, what does progress look like in your field of expertise? Sanjay, would you mind leading off the discussion?
As a scientist, progress is everything. Through scientific discoveries, we can actually create new technologies that move the world forward and address some of our world's biggest problems. In my area of expertise, we've gone from sequencing the human genome to now sequencing what genes are expressed in single cells. And this has been a huge way to discover how we form as organisms and how we came to be. And I'm really excited about this.
Thank you, Sanjay. Doan, how about if you go next?
Scientific progress matters because it's the way we learn about the things around us. If we can understand how things work, we can use this to understand and improve our way of living, especially for future generations. Without scientific progress, we would have shorter lifespans, higher mortality rates, and major global food shortage. While most of what we do as entomologists don't make it to commercial use, but we can provide information from our research to benefit growers in their day-to-day operations. This can range from changing the timing of sprays to developing a new pest management strategy. Progress to me in my field is when we can see farmers and growers benefit from our research and in turn benefit consumers like you and me.
Brian, would you answer the 2 questions for us?
So scientific progress matters because the knowledge that we gain helps us guide our decisions. It helps us understand the world around us, and it is what moves humanity forward. Progress in my area would be understanding how the brain functions, understanding what is happening with individuals suffering from neurological or psychiatric disorders, and developing therapeutics to target them.
Nick, it's your turn now.
So scientific progress is really important because it's how we develop any new therapy or new intervention that can improve the way of life that we have. And I think for my field, it's best illustrated by the fact that now immune cells can be leveraged as a new therapeutic against cancer. And I think that really understanding cell fates, what an immune cell will do once we sort of perturb it, will better help us leverage immunity as a therapeutic agent, not only for cancer, but for numerous other conditions as well.
Thank you all so much. We really appreciate your comments. I would now like to turn our attention to Rhea Kohler, who's an active Seattle Chapter ARCS Foundation member. Dr. Kohler will be speaking to us about Washington State's ability to organize a coordinated response to the COVID pandemic.
We've responded to the pandemic rather positively, and that's because we have listened to the science. It has been fantastic to see the politics as well as the different institutions, not to mention the brains that comprise those institutions, and the tremendous funding that we have had available to us to attack this pandemic from different angles. We've had individuals and groups, teams of people with different expertise, whether be it vaccine development, diagnostic development, epidemiology, statistics, the development of new and innovative immune assays. And we brought all of those together. We did it expeditiously because the funding has funded those programs or supported those programs. And it was not a matter of being faced with a pandemic and then needing to seek funding in terms of ensuring that the future generations of ARCS fellows are funded and that they continue having the resources to do the innovative research that different institutions in Washington State are providing. However, there's so much more that we could do. We really need your help. We've been so fortunate to partner with WRF. WRF has funded scholars at the University of Washington, at Washington State University, and of course at ARCS. For a number of years, they've been a consistent funder of innovative research, providing support in many different disciplines, which is what we need in order to make huge strides against this pandemic. One of the ways that WRF has come to the table is in terms of their new postdoctoral program, and they've funded investigators who are now trying to develop next generation diagnostics and vaccines for COVID-19, as well as of a variety of other noxious pathogens. ARCS has been fortunate to partner with the Fred Hutch as well. They have been amazing in terms of providing research space, the infrastructure, not to mention how important it is, all of the mentorship that they have provided for researchers that are up and coming. One of the important aspects that I think many of us realized when this pandemic hit us is that we were able to pivot very quickly in terms of developing new technologies, applying the knowledge that current fellows or past fellows performing successful research, mentoring as well the next generation of scientists. It was so important to see that we were ready simply because we have bright young minds already in place, already in labs. It's really amazing. We need to continue funding these young minds to ensure that we have future successes and future readiness to pivot when the next pandemic or other noxious thing hits us.
On behalf of the Seattle Chapter ARCS Foundation members, I would like to thank each of you for taking the past hour to help celebrate scientific research being conducted in the state of Washington. It has been inspiring to hear how our mission, Advancing Science in America, is being realized through the research being conducted by our fellows. Our remarkable 4 panelists are part of a larger cohort of fellows whose research will become the source of scientific advancement. As Rhea Kohler so eloquently stated, we must continue to invest in building a scaffold for scientific readiness if the United States is to stay competitive. As I mentioned in my introduction, our current prosperity, security, and good health is directly tied to the generosity of generations who came before, and it is now our turn to pay it forward so that generations to come can reap similar benefits. My husband Bill and I have been enthusiastic ARCS donors for over 20 years. We remain confident that our donations are being well spent and making a difference both to the students we support and knowing their research will have a positive impact on the world. Funds raised today will help support these bright, talented students and further the mission of ARCS. On your screen is a breakdown of the different ways to give. Online donations may be made by clicking the link on your webinar invitation or visit seattlearcsfoundation.org. Again, thank you for your generosity. Together, we can make advancing science in America an ongoing priority.
I feel very lucky, I have to say, to have received the ARCS Fellowship, and that's why I wanted to thank you all for funding this and for being a part of for ARCS.
It's inspiring that people like you are committed to students who desire to be involved in scientific research, and I'm really excited to see where this next journey leads.
It's a really amazing feeling to know that there's somebody out there who is so invested in my research and in my future success as well.
This fellowship has meant a lot to me, especially. I'm still in a little bit of shock that I'm at this program at this prestigious institution.
It makes such a difference, I think, in how you approach learning and work to know that you're being supported and also literally invested in.
I wanted to give a huge thanks to the Seattle chapter donors because despite of all the uncertainty right now, the ARCS Fellowship has provided stability during my move and made the whole process much less stressful.
It's exceptionally helpful in this time with COVID, and for me especially, it brought a lot of financial stability.
I hope you all stay well and stay safe, and hope to see you soon.
Have a great day, have a great week, and thank you for your support.